Hello, good afternoon to everybody. I'm sorry not to be here with you today, but the current situation of chikungunya in Caribbean and Ebola prevents me to come to ASTMH this year. So I propose to record this talk, and I hope this will be okay. So today I will talk about novel sandfly-borne flaviviruses that circulate in the Mediterranean area and in the Middle East, and it's about virus discovery, epidemiology and medical perspectives. So briefly, the flaviviruses belong to the family Bunyaviridae in the genus Flavivirus. They are all enveloped viruses, and they have a three-segmented RNA genome with a negative polarity majority. So the genome is segmented, so small segment encodes from the nucleocapsid and the NSS protein. The N segment encodes from the two glycoproteins, and the large segment encodes for the polymerase. So the taxonomy is quite complex because it's a large virus genus, and so far there are 9 species that are recorded and assessed by ICTV. These are the 9 species, and within each species there are several viruses, so a total of 37 viruses, and there are 33 unclassified viruses. Some of which have been unclassified for a very long time. For example, sandfly fever Sicilian virus was isolated in 1943 during World War II, and it's still unclassified, which is quite a awkward situation. And there are also new unclassified viruses because they are new and recently discovered, and we are going to talk about these viruses. So these new viruses are briefly presented here, and as you can see, they have been isolated in many countries like Turkey, Tunisia, Iran, Italy, France, Spain. And so when we look at the phylogeny of flaviviruses, it's also a quite complex phylogeny. So you have viruses that are transmitted by mosquitoes. You have flaviviruses that are transmitted by ticks, and this group is growing. It was long restricted to Cunivi viruses, and now there are much more viruses that are recorded in this group. There are viruses transmitted by sandflies in the New World, and there are viruses transmitted by sandfly in the Old World. So basically we talk only about the viruses transmitted by sandfly in the Old World and in the Mediterranean basin. So if we go back to 2009 and look at the sequence data that were available for phylogeny, the phylogeny was quite simple with 3 different serocomplex or species. This one groups the Naples viruses and Toscana viruses. The sandfly fever Sicilian serocomplex was constituted by a few virus isolates, and the Arbia virus that was long recognized, but with no viruses of medical or veterinary interest inside, is also very limited. Today, so basically 5 years later, the situation is quite more furnished with a lot of new viruses, as you can see, have been isolated in the Old World, and we are going to talk about some of these viruses and the efforts that are being done to isolate the virus and assess their medical importance. So I have decided to focus my talk on what has been done in Turkey by my group and collaborative group in Turkey, and we will take the example of Adana virus. The study area is in the Mediterranean part of Turkey. This is Mediterranean Sea, and so take place in the Mediterranean part of Turkey. It's just southern Anatolia, and the sandfly were trapped in the region of Adana, just here. So the trapping was done here. We also collected sera from humans and from domestic animals, both in the Adana region and in the contiguous region of Mersin. So as you can see, the total distance is quite important. It's about 200 kilometers wide, and sandfly were collected only in this region. So the result of this sandfly collection that was done by some of our students and helped greatly with people in Turkey. So the total number of sandfly collected was almost 8,000 sandfly in different locality, but quite restricted locality. I mean, it's small diameter, and they were organized as almost 400 pools to be tested for the presence of viruses and Leishmania. So the approach is always the same. The sandfly are crushed after being pulled, and after they are aliquoted, and one part is dedicated to the nucleic acid testing and PCR detection, but also now we are doing next generation sequencing directly on the pools. The second part is to inoculate the viruses, because flaviviruses, sandfly-borne flaviviruses, only grow in viruses, and so are inoculated with these pools and looked for CPE and for electron microscopy, and on this supernatant after NGS sequencing is done on viruses that have been putatively isolated. So in this case, electron microscopy on the passage 2 supernatant provided an image. We had CPE and an image in electron microscopy that is quite evoking flaviviruses or Guinia-Iridae. And we performed the complete genome determination using NGS on ion torrent machine using the de novo approach. And by doing this approach, we were able to sequence the complete genome. So for the polymerase gene, as you can see, Adana virus is grouped with Arbia and Saliabad in the Saliabad complex. The GN gene also group Adana in the same complex. Same thing for the GC gene. For the N gene, we also have the same position, and for the NS, it's also the same position. So what we can say from this rapid phylogenetic analysis is that Adana virus consistently has grouped with Saliabad species member, and so there is no question of recombination or reassortment for Adana virus. Now the second question is Adana virus a novel virus or a novel species? So if you look at the distance that are observed between Adana virus and the other Saliabad viruses, they are in different colors, each color for one protein. And the distance, the lowest distance that is observed interspecific, meaning between the 9 recognized species, the value between Adana virus and Saliabad complex is always lower than that observed the lowest interspecific distance, suggesting that this is not a new species, but that Adana virus is a new virus, a new member of the Saliabad species. So the next thing was to address the potential human and veterinary impact of these newly discovered viruses. So what we've done is to use neutralization assay by microneutralization technique with twofold division from 1 to 20 to 160. the 1,200 sera were tested against adenovirus, but also against RBR-virus, and all the sera were negative when tested against RBR-virus. But in contrast, by testing against adenovirus, as you can see, there is very low prevalence of neutralizing sera, positive sera in human sample, but in animal sample the situation is really different, with dogs that are infected with more than 10%, and rates even higher in goat and sheep. And which is very interesting is that these goat and sheep were not collected in Adana region, but in Mersin region, which is the region that is located west, and I showed on the map previously. So, although adenovirus was isolated in Adana area only, and not in Mersin area, because the trapping of sandfly was not done in Mersin area, the neutralization results show that adenovirus is also circulating in the Mersin area, so that the area of circulation is quite large. So the human results suggest that the impact on public health is likely to be limited, but merits to be further investigated. So using the same methodology, we have also isolated other strains of sandfly-borne flaviviruses in Turkey. And here you have a brief description. So here you have the region of Adana, and Mersin region is here. And in this region of Adana, as you can see, the most interesting point is the isolation triangle. And so the triangle for adenovirus is blue here. The triangle for Toros virus is green. So there is isolation of a virus that we have called Toros. And the third triangle is the triangle in yellow, and it stands for Zerdali virus, which is neighbor-like virus. So when we look at the phylogenetic tree, and the complete sequence was determined for Zerdali virus and Toros using the same methodology as for adenovirus. And I'm going to present only the sRNA phylogenetic tree, because it's the same for all other genes in the N and in the L. And as you can see, Zerdali is distantly related to Terran virus within the Toscana and Naples serocomplex. Adenovirus is in this position, and in Toros virus there are two viruses isolated, and they are closely related to Sicilian-like viruses. So for these viruses, the 1,200 human and animal sera are currently being tested for the presence of neutralizing antibody against Toros virus and Zerdali to again assess the human and veterinary impact of these newly described viruses. So the results are pending. The last point is a study that we did using again the same methodology in Iran. And in Iran we have a collaboration with Valide Moinvaziri, and she's doing a great job in collecting massive amounts of sandfly all over the country. And these collects were done in 2011 in 4 different regions of Iran, and they were tested using the same technique. So the total is almost 4,000 sandflies that were trapped in 4 different regions of Iran, and they were organized as 164 pools. So what we found is one pool that provided cytopathic effect in viral cells, and these pools corresponded to flies that were trapped in the village of Shogakesh in the Dashli-Boron region, which is here, just very closely of the frontier of Turkmenistan. And the flies were not phlebotomine flies, but they were Sargentonia flies. So here you have the phylogenetic position of Dashi virus, that is Sicilian-like virus, based on analysis done in polymerase. In the glycoprotein, the position is the same. I'm sorry, because the rectangle has shifted a little bit, so Dashi is closely related to sandfly fever Turkish virus, and in the sRNA virus is also in the same region, so no reassortment, no recombination, and human and animal sera will be collected this year to be tested for the presence of neutralizing antibody against this newly discovered virus in Iran. Collection have started now. So I think I'm done. So I was very happy to prepare this video projection, and hope that next year I will be able to join you. And so I hope your working day has been fine, and that you will enjoy this talk and the continuation of the meeting. Thank you.